Good evening to one and all. Today we are going to speak about DNA and chromosome, and we are also going to speak about more about chromosome, like some more details about it. So first of all, I would like to show you the structure of DNA. Okay, it's like this. Okay, but I will show it to you in this way, in this horizontal way. So this is DNA double helix, which is a genetic material. If you can see, it looks like a ladder. Yes. Okay, so in this these, if you can see, so these stair like thing, these are nitrogenous bases, and these ball like things are sugars. Okay, and with the sugar there is an attachment of phosphate. So speaking about DNA, it is a genetic material. Okay, genetic material means that it is like, it can be heritable. Okay, so it is passed from the parents to the offsprings. Okay, so this is genetic material. Okay, and second thing, it is heritable, or you can say that the material which is inherited is DNA. Okay, and on DNA there are genes. I'm not going to go much detail about it, but I just want to take this part as superficial. So from parents to offsprings, this is like the characters or the features, okay, which are passed from parents to the offsprings, that occurs due to the passage, like the DNA is passed from parents to offspring. On DNA there are genes, that is genetic material. Actually DNA is a genetic material. So this parents to offspring transmission is known as inheritance, hereditary. Okay, so I was telling that DNA has three parts. What are those three parts? Okay, it is made up of three parts. First is phosphate group. Second is sugar, and specifically it is pentose sugar. Okay, it is specifically pentose sugar, means 5 carbon sugar. And the third thing, which is known as nitrogenous base. Nitrogenous bases, which are two types, that is purines and pyrimidines. Okay, purines and pyrimidines. Fine. So this is all about DNA, and this is its parts. There are three parts in DNA. So DNA is made up of these three parts. Okay. So further speaking about like DNA. So DNA, the size of DNA, it is in centimeters. Okay. So it is in centimeter, means DNA is so long. But we know that our cell is in micrometer. So centimeter is a bigger unit and micrometer is a smaller unit. So the thing is very clear that something is occurring, that DNA is like converted, like it is condensed, it is packed. Okay, now I will take one example. Suppose I have small bag and I have a huge, you can say a very long sari. So I have to fold the sari to fit it into that bag. In the same way, I have, like, if we have this long DNA which is in centimeters, okay, this is in centimeters, that is to be packed like this. Okay, so if I will say that DNA was like huge in centimeters, then it was actually packed and it was looking like this. Now it is smallest, So the condensation goes to this small. So I will tell you that if DNA is too so the condensation, so this is the whole length of the DNA is in centimeters, okay, but the diameter, okay, like this size is 2 nanometer, this much, okay. So the condensation goes from 2 nanometer to 1400 nanometer, and this 1400 nanometer something looks like this, okay. This is just an example, but actually it looks like this. So this is the highly condensed form of DNA known as chromosome, which is 1400 nanometer condensed, highly condensed form, okay. So I will write here, this is chromosome, okay, and this is highly condensed form, okay, compact form, highly condensed form, highly compact form. So this is about the DNA 2 nanometer, and this, you know, this thickness will be 1400 nanometer, means just imagine it's so smaller. It's so small that in a cell, okay, this is a cell. In the cell we have nucleus. In this nucleus, the chromosome is present in the nucleus. So you can see, so to fit into this small cell, which is in, with this cell, actual size of just cell is in micrometer. So you can see that below, like this size is in micrometer. So the centimeter, the size of the DNA size, you know, the length was in centimeter. Now it has been, the thickness of this chromosome is in 1400 like nanometer, which I told you here, and this is chromosome. So I hope till here this thing is clear. Moving forward towards the next part, which I will explain, okay. Before that, you can see this, what I have written till now. Okay, speaking about the condensed form, which is chromosome. Okay, now I will speak in detail about chromosome. So, like the discovery of the chromosome was occurred when the, you know, after the genetics field was understood by the people, by the scientists during that time. So after 1009, like 1900, so during this time the genetics was understood, okay. And after that, in 1903, in this era, the, like in this year, the chromosome theory was given, okay. So the chromosomal theory of inheritance was given by the two scientists, Boveri and Sutton theory, and then they understood that what is chromosome. And the, okay, so there was a theory given by Sutton and Boveri. I will write the words first before continuing. Okay, so these two scientists gave, and the word chromosome, okay, so the word chromosome, chromo and zome, okay. So it, we can break it into two words known as chromo and zome, different. So chromo means colored and some means body. So colored body, okay, this was the, you know, etymology of this word, okay. The meaning of this word is colored body. So this is colored and this is body, okay. And the word was, this word was coined by Valdier. The name of the scientist who coined the word is Valdier, and this was the theory, okay. In 1903, the theory of chromosomal, like theory of chromosomal inheritance was discovered. Okay, so the chromosome is a colored body, and it is actually like the main function of the chromosome is for inheritance. Okay, so it carries the information, the genetic information from the parents to the offspring. Okay, so this is chromosome. So this is a chromosome. You can see that the chromosome is having two arms. Okay, two arms is there. Okay, if you will say this and this is two arms, and this is again two strand. Okay, so let me draw it here. So this is the chromosome. Okay, I will use something dark color. Okay, this is perfect. Okay, so this is a chromosome, and this is one arm and this is second arm. So this arm we say it as P arm. Okay, so this we say it as P arm, and this we say it as Q arm. Yes, but this circle, okay, this is something which is, it is keeping two this, like two strand intact. Okay, instead of saying strand, the actual word is chromatids. Now I will explain you what is chromatids. So something which is attached between these two chromatids, if you can see here the darkened part, that is known as centromere. So this circle part is known as centromere. Okay, I will make the concept clear. First I will show you what is this. See, I have taken this entire chromosome, and this is the exactly half. If you will take exact half of this, then you will get this. Okay, so this is chromosome, and just one half of it is known as chromatid. Okay, so this is chromosome, and this half of it is known as chromatid, and both the chromatids together, they are attached by a center known as centromere. ठीक है? So this was about this, like what is centromere and all. So if you take a half of this, so you will get one chromatid and second chromatid. Okay, so it will be like this. Yes, since they both are attached together by one centromere, so we say these two chromatids as sister chromatids. Okay, this is centromere, which is like, it is attaching two chromatids. Okay, let us consider this is one chromatid and second, it is attaching two chromatids, so we say it as centromere, which is present here. Okay, we can't say that it is present only in center, it can be present at any position. This also we will discuss in upcoming part. Okay, so this is one centromere, this is one chromatid, and this is second chromatid. Okay, so the actual, I told you that the thickness of this part, it is of 1400 nanometer. So exactly if I take half of this, so this size will be 700 nanometer, and if I take again this size, then this will also be 700 nanometer. Okay, understood? So the thickness will be half of it, since these two chromatids are same, and this is attached by one centromere, so we say it as sister chromatid. But what if I speak about this chromosome? Okay, so if I say this is also a second chromosome, this is the first chromosome which we were speaking about. If these two are sister chromatids, So this will be also sister chromatids together. Okay, so these two are also sister chromatid. But what about the relation of this strand with this strand? So this strand and this strand are known as non-sister chromatids. So the thing is, when one centromere is attached by two chromatids, okay, it is attaching two chromatids, so we say it as sister chromatids. But if there are two different chromosomes, and if we are speaking about two different chromosomes, chromatids, then it is known as non-sister chromatids. So these two, so this and this, we say it as non-sister chromatids. Okay, so the two words are different. This is a different word, like different meaning, and this is something different. So the sister chromatids is for these two things, or you can say for these two. Understood what is sister chromatid? So till now we spoke about what is DNA, we spoke about what is chromosome, what is chromatid, what is sister chromatid, what is non-sister chromatid, and what is centromere. Okay, you can see this, have a look towards this, and then forward we are going to speak about the next part, that is what is homologous chromosome. Okay, I hope that till here this is understandable. So today next part we will take it, that is homologous chromosome. Homologous chromosome. And second I will take as non-homologous chromosome. Okay. So the two chromosome, okay, if there are, this is one chromosome, yes, and this is the second chromosome. So these are two chromosomes. So during the cell division, what actually happens in meiosis? In meiosis, during the cell division in meiosis, that also in prophase 1, and in the prophase 1 in the zygote, the homologous chromosome comes and they pairs. One chromosome comes from the paternal side, that is from the father side, and one chromosome comes from the mother side. Okay. So to make it very clear, let us take two chromosomes. Okay, I am taking two chromosomes. Blue color is from father side and pink color is from mother side. Okay, blue color is from father side, pink color is from mother side. So during the meiosis occurs in the germ cells. Okay, so during meiosis, one chromosome is from the paternal side and one chromosome is from the maternal side. Okay, so this is the process where the zygote, we will speak about the zygote formation and all. So homologous chromosome is there. So if you can see that these chromosomes, they are exactly identical to each other. Now how they are identical? If you can see the size, okay, so if you can see the arms size are exactly same. The centromere position, if you can see this centromere position and this centromere position is exactly same. Even this P arm and this P arm are same in length and size. This Q arm and this Q arm are same in length and size. So they are structurally same. Morphology means what? Outer appearance is same. Okay, structurally, morphologically they are same, and even genetically they are same. Okay, means the genes and all, they are same. So these three things, structurally, then morphologically and genetically, they are identical to each other. They are identical, means exactly same. So they are identical to each other. That is what we say it as homologous chromosome. Okay, so this is what we say it as homologous chromosome. So they are structurally, morphologically, okay, it is structurally. I told you how structurally. If you can see here again, or you can even see this two chromosome pair which I have drawn on the board, or you can see this. Structurally same, morphologically same, and the genetically same. Okay, so the first one is structurally same, second is morphologically, and third is genetically. They are identical. What are they? They are identical. Such chromosome pair are known as homologous chromosomes. And these chromosomes pair during meiosis 1. Okay, meiosis 1, and that also prophase 1. In prophase also zygote. Okay, I will take a soon. I will bring one video. We will speak about cell division also. But for now, you should know this much. Okay, understood? So this is homologous chromosome. But now there are some chromosomes, like if I will take X chromosome, if I will talk about X chromosome, so X and Y chromosome. So these are sex chromosome in humans. Okay, so X chromosome is somewhat like this, and Y chromosome, if I say like this. So if you can see that, if you can see that this much part of both the chromosome is same, but the above part, if you can see, the upper part is not same. Okay, if you can see the upper part is not same, मतलब ऊपर का जो पार्ट है वो साइज में सेम नहीं है, नीचे का जो पार्ट है वो साइज में सेम है, मतलब स्ट्रक्चरली एंड मॉर्फोलॉजिकली दे आर आइडेंटिकल. So the lowermost part are known as homologous part. But the part which is not structurally same, not morphologically same, and neither they will be not genetically same, means they will not obey these three things. So we say it as non-homologous part. Or you can say not homologous chromosome. Non means they are not structurally identical, nor morphologically identical, nor they are like genetically identical. Understood? This is about homologous and non-homologous chromosome. The further difference about non-homologous and homologous chromosome are the crossing over. I will speak about a crossing over concept. Soon the concept will be clear. Sorry. So homologous chromosome, they undergo crossing over. Okay, so these chromosomes undergo crossing over. Okay, but these chromosomes, they don't undergo no crossing over. Okay, they don't undergo crossing over. So the main criteria for undergoing crossing over is that the two chromosomes must be homologous chromosomes, that is, they should have structurally, morphologically and genetically identical. They should be. It's compulsory, it's mandatory. So here there will be no crossing over because it is non-homologous chromosome. But here in homologous chromosome part there will be crossing over. Okay? Now what is crossing over? Since when I am saying crossing over, but what it is? So I will speak about it. Just have a look towards this, and then we will move forward. Okay, so now we will speak about what is crossing over. Okay. Without writing anything, just I will put the heading that this is a crossing over. We can also, okay, just write crossing over. Now I will just explain some part. Suppose this is one chromosome and this is one chromosome. 1 second. Blue one is from the okay, I will take this red one to give you a good idea. See, this is blue chromosome which is from and this is the thing which will depict blue chromosome, and the pink chromosome will depict this red pen. Understood what I told? This is paternal chromosome and this is maternal chromosome. Okay. So these are the chromosome and we say it as homologous chromosome. So I told the first condition that crossing over occurs in homologous chromosome. Okay. So when a part of paternal chromosome, it's detached, little small part, and from maternal chromosome it is detached, okay, then it is attached to each other. Like, you know what happens, little part comes from the paternal side which is attached to the maternal chromosome, and little part which is attached to the maternal chromosome is back attached to the paternal. Okay, I will show you on the board what actually happens. Okay, so this is one chromosome and the second chromosome is this. Okay, I think wait one second. Okay, so this is from father side and this is from mother side. Okay, now what happens? First they came together and they will attach. Okay, they will come together and they will attach. How they will attach? Like, this is the mother chromosome, the chromosome of the mother. Okay. And the other chromosome from the father side. So if you can see that both the chromosomes are coming and they are attaching to each other. I have shaded that part, the part which is getting attached. Okay, I will shade it with this. Okay, so they are attaching here. Yes, so they are attaching here. So they have holded each other. Okay, so holding will be in this side. It won't be at this and this side. Okay, now suppose if I am here, my friend is here. So we will hold hands by one side. We can't hold the hand from both the side. Okay, so it's just about this position. Don't tell me that we will hold like this. It's not about that. We will just hold one side. Same way the chromosomes are also holding this way, and what happens when they have holded, so they exchange their parts. Like the way I was showing this to you. See. So I told that they came, they holded the ants and they exchanged their parts, and this is what something new formed, a new combination is formed. Okay, so this is a new combination. Earlier it was like this, and now there is a formation of new combination. This new combination is known as recombination. Okay, and this is what is known as crossing over. Okay, so new combinations were made, which leads to formation of recombination, or you can say which this is actually a recombination which I am showing you here like this. So these are recombinants, something new has formed. Suppose if I take white color and I took pink color, if I mix those color, then there is a formation of pink color, which is a new combination, which is a recombination of two colors. Same way, as the chromosome part of paternal and maternal, they exchange little part, not full. Then it is known as crossing over, and it is very requirement. It's a very essential, I must say, for the variation. So this brings variation. Now what is variation? Just you stand in front of your parents. You and your parents, you three are not identical, or you can say you are not identical as your parents. Why? Because this is a sexual reproduction. This is it's not like a sexual reproduction. So my face and my parents' face will not match. I'm not an amoeba, not they are amoeba. We have not undergone, they have not undergone a sexual reproduction. So when there is in humans, there is a sexual reproduction, which brings about crossing over, which brings about variation. Variation means some unique features will be there. Okay, when you have observed, when a new child is born, we say that this feature is from mother, this feature is from father. Okay, we don't say that entire it is of mother or entire it is of father. The child is a combination of father and mother's genes. Okay, and there will be something new, a recombination that will bring variation, that will bring uniqueness in the child. That is known as variation. Okay, so this is about the entire topic which I wanted to explain, the crossing over process. I will show you crossing over by the help of this model. Okay, so this is crossing over. Okay, so first they will pair in meiosis 1, both the homologous chromosome they will pair. Afterwards, as they will pair, they will attach like this. Then there will be this thing. Then there will be what? So this was the first condition, and this is the next condition. So see, this was just pairing and holding each other, and then there will be crossing over. If you can see here that this little part has came from maternal chromosome, pink color chromosome, towards the paternal blue color chromosome. Here also if you can see, and the maternal chromosome is having blue part of chromosome from the paternal side. This information which I told you till now, this is a very important concept. This is known as crossing over, which brings about recombination, which is also going to give you variation. I think this is very clear. Okay, so I spoke about chromatids and all. So I will just show you once again this crossing over concept, so that it will be clearly understandable to you. Okay, just I will come forward and just I will show you this. So if you can see this, so see these chromosomes. Okay, and then you see these chromosomes. Okay, I will remove my hand. Just see this. You will get the understanding of what I was actually trying to explain. Okay, so I think this is clear. Moving towards the last part of today's class. So today's class, last part I will explain, and then we are going to end this class. Okay, so you can see that whatever is written on the board, you can just take it down if you want, and then we will move forward. So this is done. The last part we are going to speak for today's class. So types of chromosomes we are going to speak. Okay, the topic is types of chromosomes. So this topic is divided into two parts. First part is based on like which type of chromosome it is, like whether it is autosome or sex chromosome. Okay, so the first part is autosome and sex chromosome, and the second part is based on position of centromere. Okay, so based on the position, like where the centromere is present. I told you what is centromere. So based on it, there are four parts. First is metacentric, and second is submetacentric, and the third is acrocentric, and the fourth one is telocentric. So in human, if I say there are 23 pairs of chromosome, out of those 23 pairs of chromosome, there are 22 pairs of autosomes and one pair of sex chromosome. So the sex chromosome, it actually determines the sex, or you can say the gender of a human. If it is girl, then it will be XX for a female, and for the male it will be XY. Okay, so they are actually determining the gender of a human, like whether it is male or female. Speaking about like based on the centromere, now I told you what is the centromere, which is which is holding two chromatids. That is a centromere which is present. Okay, the position is not fixed, whether it is in center or what. Okay, so based on it, there are four types of chromosome. The first is metacentric. Metacentric means the centromere is present at the center. Okay, if the chromosome, the centromere, if it is present at the center, then we will say it as metacentric chromosome, because it is present exactly at the centromere. Okay, I can show it like this also. If the chromatid I will take, then it will look like this. Okay, so if I take this structure and if I try to fold it, or you can say like, if I want to see its shape by folding, then it will form a shape as V. How V shape? I will show you. So this is a metacentric chromosome. Okay, if this is a centromere in middle, if I want to fold and see the shape, then it will be as V shaped. Okay, I will come in forward, and I will show you. This is centromere. This centromere of this chromatid is present in the center. If I want to fold it like this by the centromere, then this will be known as a V shape. So I said that this metacentric is a V-shaped chromosome. Okay, it is a V-shaped chromosome. Yes, understood. So this is a V-shaped chromosome. Okay. Speaking about the next chromosome, which is submetacentric, means from the center से थोड़ा सा ऊपर चला गया the centromere. अगर center से थोड़ा ऊपर चला गया, center से, this is center, so if it went up, we can say this is also not center. If it is like exactly at the center, okay, then we can say it as a metacentric. If it shift little bit above the center, then we say it as submetacentric. So I will again come forward and I will speak about the shape. So it forms L shape, and this is submetacentric type of chromosome. If I want to fold it, it will look like this, and it will form a L shape. Which shape it will form? An L shape. This is submetacentric and it will form an L shape. So we say it as, like, the shape looks as a L shaped. Okay, it looks as a, okay, now the pens have stopped working, they are also tired, I feel. So this is L shaped. Okay, L shaped chromosome. So speaking about the next chromosome type, or you can say based on the centromere, that is acrocentric. Okay, acrocentric means एंड से थोड़ा नजदीक. ठीक है, सेंट्रोमियर कहाँ प्रेजेंट है? एंड ऑफ द क्रोमोसोम से थोड़ा नजदीक प्रेजेंट है. इट विल लुक लाइक दिस. यहाँ पे एंड ऑफ क्रोमोसोम से थोड़ा नजदीक प्रेजेंट है. ठीक है, आई विल ड्रॉ इट अगेन. तो ये जो एंड है ना क्रोमोसोम का, उससे नजदीक प्रेजेंट है, इसके लिए हम बोलते हैं इसको एक्रोसेंट्रिक. Okay, see, I will show you. So this is a chromosome. ठीक है, this is a chromatid. Okay, I have not shown two bands, just I am showing you one chromatid and explaining you. If you can see that the centromere is here, it is towards near the end. So this is end of the chromatid and it's near the end. So this centromere is near the end of the chromatid. So we say it as acrosome. And acrosome looks like a J shaped. So if you will do like this, okay, something like this, if you take it as up, so it will look as a J shaped. So acrocentric looks like a J shaped. Okay, it looks like a J shape of a chromosome. J shape. Last about telocentric, the centromere will go at the end of a chromosome. Means at the end it will be attached like this. If you will see here, the centromere here, it is at the end, exactly at the end of the chromatid. If you take two chromosomes, then also it will be looking like that only. Means take one more. Okay, if I take two chromosomes, so it will look like this, two chromatids, sorry. So it will be at the end of the chromosome, like this, centromere. Okay, so this is telocentric and it looks like I shaped. Okay, so it looks like an I. If you want to see this, see, it's at the end. The centromere is at the end. So this is about all the types. Okay, and I will show you once again. So these are the types. See, the first one which I have shown here, it is submetacentric, and the second is metacentric, and this is, okay, the first one is acrocentric. Okay, I told wrong. This is acrocentric, this is metacentric, submetacentric, and this is telocentric. So you can see it well, I think. Yes, you can. Okay, so I hope that today's class you find it useful, and today's things you would have understand also. So this was my side. This was from my side. But last thing which I want to say, which is very important. So during a cell division, okay, if there is homologous pairing, okay, homologous chromosomes are attached, okay, so it's like this, they were attached, then at anaphase 1 they will segregate, at anaphase 1 they will segregate, they will separate. During anaphase 2 of meiosis 2, what will happen? If there is paternal chromosome, its centromere will separate. If there is maternal chromosome, then its centromere will, or you can say that it will separate by its centromere. Okay, if I say this is paternal chromosome, then it will separate like this. Okay, it will separate like this. There will be a pull. Okay, there will be a spindle fibers pulling upwards, there will be a spindle fibers pulling downwards, so it will be a pull, and this way the chromatids will separate. So once again, during anaphase 1 the homologous chromosomes separate, during anaphase during anaphase 2 the chromatids separate. Okay, once again, if you were not able to see, so during anaphase 1 of meiosis 1 the homologous chromosomes, okay, I will put it properly first of all. See, it's like this. The position should be like this because crossing over had occurred here. It will be here. It will occur only towards the holded position, means where they were attached. So during anaphase 1 of the meiosis 1 they separated, homologous chromosomes separate, and during meiosis 2 anaphase 2 the chromatids separate. Okay, means the paternal chromosomes chromatids will separate and the maternal chromosome chromatids will separate, and this will be the cell division. Okay, so this was about today's idea, today's concept. I hope you understood these things. At last, I would like to give you a short summary what we have studied today. So we spoke about DNA, what it is actually and what all it consists of, what is the function of a DNA, and then we spoke about how the packaging of DNA occurs to till the chromosome, second thing. Then third thing, we spoke about what is chromosome, what was the discovery and all, who discovered and everything, the theory and all. Then the fourth thing, we spoke about the types, sister chromatids, what is sister chromatid, then what is non-sister chromatid, what is homologous chromosome, non-homologous chromosome, what is crossing over, and lastly we discussed this types of chromosome, autosome, sex chromosome, and the based on where is the position where actually the centromere is located. So this was for today. I hope you found it very useful. So thank you.